Good morning. Good morning. This is a really fun day for me. My name is Rammonhan Smith. I'm the chair of the Department of Neurosciences here at the University of Toledo College of Medicine and Life Sciences. I started back on August 1st. And one of the things that I've been involved with now for 3 or 4 years is becoming what I will call an educated user of bioinformatics. I'm not a bioinformaticist. I've learned to sit in the room with bioinformaticists and be able to communicate with them without losing my mind. And that's not a new joke to Dr. Miller, who's been a phenomenal colleague, collaborator, coach, scientific friend. And the idea for the mini symposium today really came together with the co-chair of the symposium, Dr. Isaac Schiefer, who's down here in the front row, to bring together the folks that are thinking about different ways to approach drug discovery, repurposing drugs, not just at the level of, hey, let's find a target and give it to an animal model, but all the way back, starting with the basic science as well. And that's really the part of the sort of the big overall arching pipeline of drug discovery that I'm involved in, is identifying pathways that could be targeted. I'm not somebody that runs clinical trials or does high throughput screening or anything like that. Stuff I'm interested in. So that's kind of how this came together. And today we have a wonderful roster of folks that I guess I would call scientific friends of mine that I'm thrilled to bring together and share with the audience. Of course, today we have people here in the room. We're webcasting to folks all over the country. We're recording today's sessions so they can be viewed later on the web. And the goal is to have this be an annual event and grow it as indicated. Just to give you a quick overview of what we're going to do, we're going to have a series of 20-minute talks. I've asked each speaker to tell a story and not go too far over our heads, because many of the people in the room are not well-versed in all of the granularity of the things they're doing. And that's going to take about 2 hours. And then we'll have a biological break for a couple minutes. And then we're going to demonstrate some of the software tools that the speakers are talking about, running actual simulations of the software in real time. So this is intended to be somewhat informal, and there is some structure, but you're welcome to ask questions. My assistant in the back, Michael, has microphones, and if you're going to ask questions, we'd love to have you do it on the microphone so that the folks listening remotely can hear the question. Alternatively, the speaker can repeat the question. So we have 5 speakers today. The first is going to be Dr. Shukla, then Dr. Miller, and then we have a bit of a typo on our program, despite spending 6 weeks working with the marketing folks to get it right. Doctors Regal and later on Dr. Alganam are not doctors yet. They were excited to hear that we've already conferred their PhDs. But they're actually still in training. So I apologize for those typos in advance. If you were sucked into coming to the mini symposium because you saw that they were doctors, then that was fake news. But we're going to go ahead and get started. And I'm going to tell you about our first speaker. Let me get to the right notes here. Dr. Rammonhan Shukla. Dr. Shukla is a new recruit to the University of Toledo College of Medicine. He's joining my laboratory group, the Cognitive Disorders Research Laboratory, where he will be in charge of the bioinformatics pod. I think of him as a bioinformaticist. He's got a PhD from the National Institute of Basic Biology in Japan. He's also got a master's from Tamil Nadu Agricultural University in India. He has spent a number of years as a postdoctoral fellow at the University of Toronto, studying gene expression changes in postmortem brain with Dr. Etienne Sibel, who's one of the leaders in the field. And Dr. Shukla and I have been working together even before he started, and he's really been phenomenal in helping us get our heads wrapped around, at least in my laboratory, at least one way to look at RNA-seq data. And I think he's going to tell you something about that today. So we'll go ahead and start with Dr. Shukla.
Yeah.
You need to hit the... Yeah, because we need to get it on the screen.
Yeah. Thanks, Rob. Thank you. So today I'll be presenting my ongoing project on discovering drugs for causal pathways associated with major depressive disorder. And to start with, I'll give you a brief outline of today's talk, what I'll be presenting today. So at first, I'll be explaining you the disease trajectory, how the disease trajectory flows. And then how the data which I'm working with fits into that disease trajectory perfectly. And with that data, we can actually segregate the trait, state, phasic, and like progressive changes associated with the major depressive disorder. Next, I'll be focusing upon developing concept using like the pathway analysis of how changes are associated with these contrasts in respect to depression. And finally, it's always good to have a validation. So I'll be showing you three kinds of validation. One is like a technical validation using qPCR, cross-platform using same data, same cohort, proteomics data, and cross studies like other people have came to the same conclusion. Our final aim, though, is to discover drugs. And in this final version of my talk, I'll be discussing my pipeline for drug discovery. But before that, there is one important thing that there are two kinds of things. So like when you have a differentially expressed gene profile or a pathway profile, those can be associated either with the cause of the disease or with the effect of the disease. And when we target some pathways which are associated for a drug discovery pipeline, we need to target those which are cause of the disease. So I'll be showing you some network analysis, Bayesian network analysis approach which I have used to target the causal effects of the disease. So let's start with the disease trajectory. In terms of severity symptoms, with first episode, there's an increase in severity, which after the treatment and into the second episode, you can see that the severity increases. And with severity, you can also note that the time spent in the episode also increases. In the subsequent remission, the remission is very small. And you can see that the time spent in the remission is very small subsequently. And in the final stage of the depression, the treatment actually doesn't work. So I was very fortunate to get the data from all the phases of depression from Pittsburgh Brain Donation Program. So I have like 20 control, 20 first episode, 20 recurrent episode, 15 and 15 for remission each, right? So I extracted the RNA from subgenial anterior cingulate cortex and performed the RNA-seq. This is like the bioinformatics pipeline. I performed like 2 into 100 base pair, reference genome was GRCS38, HiSat2 aligner. Normalization was done using HiSat2, DSeq2, and was controlled for age, sex, and PMI. Now, there are various things you can ask using this present data set. Okay, so based on these present data set, I had 7 contrasts, which can answer me what is the trait associated change with depression. So that goes with like control versus everything in MD. The second was state associated changes with depression, and that goes with like control versus the change in episode. The third was control versus the remission state. The fourth was comparing only the remission versus the episode. And this is important, like what are the progressive changes in episodes? What are the progressive changes?
Quantify this sinusoidal curve. Is it serotonin or
What is that? So the final one is actually the sinusoidal curve, and I have developed a model. I have fitted the model for the sinusoidal curve for that.
but is it serotonin or what do you measure to quantify? Do something that's manual.
So this is all like transcriptomic profile,
Yeah.
RNA-seq, right? So the final one is the phasic changes associated with this trajectory. And now coming to the result, before coming to the result, I want to pinpoint like 2 important things about the subgenial anterior cingulate cortex. It is well known that subgenial anterior cingulate cortex is more of a hyperactive disease, shows a more hyperactive disease state. What I mean is when there is an episodic state, there is like a hyperactivity in this particular region, and when there is a remission, the hyperactivity is goes down, right? And the second one, which I have already mentioned, is the disease trajectory follows this sinusoidal curve. So proceeding with the data, first of all, I looked at these two patterns in my data. So clearly, if you see the data, the remission ones, this and this, the contrast including the remission, they are like having both up and down regulated same, but all the episode stage had more upregulated gene profile, suggesting that there is a probably hyperactive disease state. And the second is you will indeed see that there is some kind of a phasic trajectory in all these states, like up, down, and up, suggesting that the gene expression profile from subcenial anterior cingulate cortex actually fits into the disease trajectory. So I did various other ways to confirm that the data fits into the disease trajectory, and this is one of it. So if you see the remission and progressive remission versus the progressive episode, one can imagine that the trajectory are totally opposite. And likewise, if you see the overlap between the differential expression profile, both up and down, there are minimal overlaps, suggesting that the actual data is fitting into this trajectory. So once I got the differential expression profile, my next aim was to target the pathways associated with these, right? And what I did is for all the contrast, I made the list of differentially expressed genes, rank ordered them, and found the enrichment of gene ontology terms using GSEA pipeline. By end of the analysis, you will be getting activated and deactivated pathways, which are actually the enrichment in the up and the downside of the rank order list. And then this is the result. Actually, there is many things to discuss in this particular slide, but I'll tell you what, like all these rows are actual one pathways, and one column is associated with the contrast. And what I have also done is I have associated all the pathways with some kind of theme, which is from central dogma till the core innate immunity, right? So segregating all the pathways into central dogma till immunity. So there are many things, as I can say, like many things to appreciate in this slide. One thing is like all the transcription related pathways are upregulated in depression in all the episodes, all the phases, whereas the translation related pathways are downregulated. If you look at over here in the core regulatory pathways, so both the state and the trait are upregulated with the core regulatory pathways, including like the matrix, inflammatory response, cytokine response, growth response, and whatnot. And if one can expect that when there is a remission, there is like a recovery phase going on, right? And in the remission state over here, you can see that the pathways associated with lysosome, vacuole, cytoskeleton, axon, dendrites, and synapses are overly increased. And if you compare only the episode versus the remission, you can see that in episodic stage, there's a highly compromised pathways associated with neuronal activity. So again, there is a lot to appreciate in this slide, but the bottom line is disease state mainly leads to altered core cellular process, whereas the remission state mostly affect pathways associated with cell-to-cell contact and signaling. So next, like this was the result, and I wanted to validate it. This is the validation using qPCR. So you can see that the qPCR result and the RNA sequencing result had an absolute value, like correlation of 0.7. This is like I selected few differentially expressed genes, which were also the leading edge for given pathways. So this side is the result for the qPCR, and this side is the result for the RNA-seq, and you can see that both are following the same trend. So this is good validation, but remember that this can only be a technical validation because both the RNA samples are coming out of the same tube, right? So the next validation which I did is the hypergeometric overlap between my RNA sequencing studies and the same studies with the same cohort done by the proteomics analysis. The overall relation was in the theoretical limit of 0.03 of between protein and RNA. And let me walk you through these little dots over here. What these dots represent is the minus log 10 p value of the overlap between my proteomics results and the RNA sequencing results. So there was like a huge amount of like overlap between the two results. And finally, the validation using the overlap between my study and somebody else's work using the same approach. So this particular work didn't have the remission stage. So they had a comparison only between control and episode. And you can clearly see that the overlap between my study and their study is only focusing on episode but not on remission. So this overall suggests that like my data generation and analysis is going on well. So next, I would like to have some water. So next, so far we have seen the pathways associated with the disease. We have seen the gene expression profile. We have seen the validation. Now the next question is, we have to target those pathways which are associated, which are the causal of the disease. And let me tell you how this analysis originated. So last time when I visited Toledo, I believe there was somebody in the audience, Joshua Parker. Yes, he's there still. And he asked me, you have done this analysis, but how will you segregate the cause and effect? So this is one of the approach, which is called the Bayesian network approach. And for this, I used WGCNA. And so what is done, this is the simple flow chart. How it is done is you create a consensus module, which is like common across all the 5 control, like remission, all the 5 data sets which I have. And then I make a consensus eigengene. And from that eigengene, I can create a Bayesian network module. So I cannot go into deep of this, but I would really suggest going through this paper. So this is a brief of it. So what is actually done is the WGCNA clusters the gene based on the individual, like common variability. And the first principal component of a given cluster is called the eigengene. So when you correlate the eigengene, you can get an undirected network. But if you have something called sink or a source, which is associated with a prior probability, you can actually give direction to the network. So this is how the network looks, and I will briefly walk you through the network. Okay, so these are the modules, and these are the pathways associated with each modules. These are the minus log 10 p-values of the associated pathways. So the network is theoretically very perfect. So for example, module 5 is connected to module 2. Module 5 is enriched in systems development, whereas the child term, module 2, is associated with nervous system development. And the thickness of this arc determines the probability of connection. So again, like for example, module 21 is associated with defense response, and the child term module 10 is associated with response to cytokines. So it's like theoretically very consistent. So I further validated my Bayesian network approach using 3 approaches. So one is I saw the enrichment hypergeometric overlap of all the 7 contrasts which I mentioned for each of these modules. And you can see that module 10, which is highly associated with the disease state, was enriched in all the 7 contrasts. And the second is, we all know that it's becoming quite well known that microglia astrocytes are well responsible for the disease state. And when I saw the enrichment of all these associated disease, you can clearly see that astrocyte, microglia, and oligodendrocytes are highly enriched in module 10, which is associated with the disease. Likewise, it is also known that layer 1 is associated, layer 1, the anatomical layer, is associated with the disease state, and you can see that highly significant association of layer 1 with all these 3 modules. So the bottom line is module 10, 13, 21, which are enriched with defense, immune, and cytokine response, are associated with the disease state. And this result of mine exactly fits with the various theories of depression, which suggests that inflammation and immune response are responsible for the disease state. Now, all these is done. Now I'll actually come to the discovery pathway, like the drug discovery pathway. So it all started with a connectivity map from the Broad Institute. So I won't be going into much detail in this. So the connectivity map, what they did is they recorded the expression profile in various cell types against various drugs. And later on, this project was enhanced by NIH. And right now, what they do is they screen for 978 genes, which they call as the landmark genes. The technology is called L1000 gene technology. And using this 978 genes screened in around more than 1,000 cells, they see the expression profile of more than 40,000 molecules, right? And I'll just walk you through how the query works. So you give the drug against a cell line, and you generate an expression profile. In this cartoon, there is only one profile shown, but there are a million other profiles. And then you query your expression pattern against the up and the down regulated ones. Now the query can be anything. It can be either a differential expression gene list, but in my case, I have shown that module 10 is associated with the disease, so I went with taking module 10. And then if it matches with the profile of the drug, then you can expect it to be having a pro-disease effect. But if it inverts the profile, you can expect to be having a therapeutic effect. Right? So this is the search domain of the CMAP. So this is a bit old figure, but I'm pretty sure like it's still consistent. So what it shows is like the 47% of this million profile, they know everything about it, and they call it the touchstone. Whereas like the 53% is still undiscovered. Undiscovered means like the structure of the molecule is known, but the function and all is not known. So this is the result of my analysis. I'm very sorry that I had to hide the actual drug which I found because my boss is going through some patent process. And let me walk you through this result. So this is one drug. The whole column is one drug. The whole row is one drug, and the whole column is representing one cell type. So these are the 9 hallmark cancer cell lines, and these are the neuronal cell lines, right? And the red color suggests the pro-disease effect, whereas the blue color suggests the therapeutic effect. And the color is the percentile, 90 to 100 percentile of enrichment score. So it's not that there is nothing here, it only means that the percentile is low over here, right? And there's one clear pattern in this, like you would really appreciate it. Like the neuronal cell types are like totally different from the cancer cell lines with respect to the disease profile, right? And this is something like once we get the disease, we want 2 other things out of it. First thing is whether the signature coming out of it is like good, what is the quality of the signature? And the second issue which we want to go through is how good is the penetration of the drug into the cell. I'm talking only about the cell, but blood-brain barrier is a totally different game, right? So what this is transcriptional activity score. So I'll walk you through what it is. So what I did is like So this is the signature. On the y-axis is the signature strength, which means for a given drug molecule, how many landmark genes are differentially expressed, right? And on the x-axis is the replicate correlation, which means for a given drug, what is the correlation between the signatures. And then what I did is I took the geometric mean between these 2 signatures. And you get 4 quadrants out of it. So if the drug profile lands somewhere here, it suggests that the profile is having strong and reproducible effect. If it lands somewhere here, it shows that it's subtle and reproducible. Whereas here it means inert, here it means noisy. So this is the profile of clozapine, one of the drugs which is known associated with depression. And this is like one of the validation of my result also, because the top hit was clozapine, which is associated with depression. So one interesting finding was all those which are associated with therapeutic response are having very subtle changes, whereas those associated with like pro disease, they are having like highly reproducible effect. So this is like just another validation of my result. You can see that clozapine, which is like on the top hit, is already known that it is associated with curing depression. Likewise, chlorpromazine, which is also associated with curing depression. So this is just a validation that the analysis which I'm doing is going good.
It's interesting, that clozapine has a therapeutic effect in cancer cells. It's got all the blue.
It's having blue, but you see like there are few red and few which haven't crossed it, you know.
The clozapine is all blue.
Yes, yes.
Very interesting, so we never thought it's going to have an effect on cancer cells.
There we have it, yeah. So now we know that like we probably know the mode of action of most of the compounds. For example, people know the mode of action of clozapine and mode of action of chlorpromazine. But if we discover a new drug, we might want to know a mode of action of the drug, right? So this is one of the approach wherein what you do is you can create a profile. Like this is all, each dot is a signature from each molecule, and you can cluster that, and then you can overlay the cluster with those profile for which the mode of action is already known. So for example, if this cluster is known to have glucocorticoid receptor antagonist, so anything coming into that cluster, you can associate a tentative mode of action that this drug is particularly associated with this particular like glucocorticoid response. So for my particular analysis, I'm going with this particular drug, and I am trying to make that Tesny plot. This is not yet complete, so I will not go further, but because I have to overlay those drugs for which the mode of action is known. And this is the final like conclusion kind of slide for me. So we also want to confirm, right? For example, we know that the module 10 is associated with the response to cytokines. So the other thing which we can validate is the particular drug which I'm interested in. If you take the signature and see the gene ontology of that particular signature, you can see that response to cytokinin, immune system response, all these are like the top hits. So just a validation that the analysis is going well, right? So far I have reached till this point, and the next step is to perform like the animal testing using the behavior which people in my lab back in Toronto are performing. So I'll stop over here, and before that, like I will acknowledge like people doing the behavior. These are the people doing behavior. Cluster analysis, Leon French and Andy Wong helped me in this. And RNA sequencing was performed by the core facility in the CAMET. Etienne Sibyl is my boss. I'll stop here and if you have any questions.
You said when you looked at the transcriptomic signature by RNA-seq, and then you validated that by qPCR, have you looked at the protein profile?
So as I told you, like somebody in my lab has already done oh, it's not showing here. It's showing here but not here. So somebody in my lab already performed the proteomics analysis of the same data set, same cohort. So I'm not including this in my present paper, but like this is one of the aim. We have to compare the proteomics result with the RNA-seq result. This is the periphery, like very outer. You can see that the correlation between them is very high. But did I answer the question?
Okay. So sometimes qPCR you see upregulation and no change in protein.
Okay, yeah.
Sorry. I mean, sometimes you see a change in the RNA profile,
right.
but there's no change in protein.
Right.
So you need to make sure that it, oscillates along similar.
Exactly. I got your point. Yeah, but so far I have went this far, but I haven't performed any proteomics.
Any other questions?
Very nice talk. What was your cutoff for the transcriptional activity score? Was it
0.2.
done manually?
0.2.
So was it done manually or you?
So 0.2 is suggested by the paper by Subramaniam et al.
Okay.
So I straight away took that cutoff.
Okay.
I had a quick question for you. And just a pitch. So we have a couple of posters in the back. One on the left was generated with help from Dr. Shukla and has a gene set enrichment analysis if you ever want to peek at it. It's a graduate student, Marissa Smale, who's in attendance today. And one of the things Marissa's been working with you on is leading edge analysis.
Yeah.
Can you tell us what that is? Because I'm still not sure I know what
Okay,
it is.
so-
I'm pretty sure a lot of people in the audience don't know either. And you mentioned that you used that to identify the genes you were going to validate.
Yes.
So you've got an RNA-seq data set, you found pathways. How do you decide how to do a technical validation? You use leading edge analysis.
Yes.
What's the rationale for that? That's the question.
So I'm very sorry that I don't have a figure for leading edge. So you can assume a leading edge is, for example, if there are a set of genes responsible for a particular pathway in my study. So there are around how many are genes which drive that pathway in my study. And those genes are called the leading edge. In GSEA, it's not required that the leading edge may be differentially expressed. But 60% of the time, the leading edge is also the differentially expressed genes. Right? So if I had something can I draw somewhere?
I don't know if I have any way for you to draw. Just give us an image.
So have you heard of AUROC? AUROC curve? So when you I'm trying how to explain it.
You could use a mouse on the computer. You might be able to use it as a pointer. Right? Presentation mode, if you
I don't know if it's going to let you. But
It should. Right.
that guy.
That many. Generally what? Then you could draw something.
If you're able to do that with your hands.
Yeah. So what happens is you arrange your So you arrange your gene set in rank order. So this is the rank order gene, right? And then what you ask is, for example, a given pathway stress response. So you ask whether the stress response is either enriched in this area or this area. That is the basic question you ask when you do gene set enrichment analysis. And what it does is it makes a random walk, and then it takes the first gene over here. And if that matches to the gene set, okay, it keeps on scoring like this. If there is a match, there will be a point. If there is a match, there will be a point. If there is a match, there will be a point. So likewise, and then it starts to go down. So to the point where the enrichment is maximum, right? All those genes included to the point where the enrichment is maximum. It's
and
called
those are
leading
the
edge.
ones you do PCR and validate.
Yes.
Thank you. Thank you for a great talk.